Our speaker today is Bob Schindelbeck. He's an extension associate in the Department of Crop and Soil Science at Cornell University with responsibilities in research, teaching, and extension. Bob coordinates the Cornell Soil Health Laboratory operations and has been part of the Cornell Soil Health Team's development of soil management strategies to address laboratory-measured soil constraints. He's produced a number of peer-reviewed and extension articles and has delivered over 200 presentations related to tillage, soil compaction, and soil quality at farmer twilight meetings and professional conferences. Bob's going to talk to us today about soil health management. With that, Bob, I'm going to turn the program over to you.
Thank you, Jeff. I hope everyone can hear me okay. Right, so 2 weeks ago, I kind of introduced the Cornell Soil Health Test in a presentation called Applying the Cornell Soil Health Test to Berry Production. And I'm going to build on that today. The title of today's presentation is Understanding the Soil Biological and Physical Properties with a special emphasis on the management. So, move forward here. So, when we talk about any sort of soil management, we find it very useful to break the soil into, if you will, these great groups, the physical, biological, and chemical properties. The reason for that is that soil ecology itself is exceedingly complex and full of so much interaction that our only chance is to use the reductionist approach of science and kind of understand the little bits and at the same time realize we're going to have to put those little bits back together into the whole ecological package. So that's kind of the strategy that we use for understanding the soil itself as scientists. Then we take that sort of knowledge and strategy and then manage the soil to manage these different properties of the soil then to get the sort of interactions moving in the direction we want. So the power of management or wise management is understanding what's beneath it. So we kind of model that strategy with this with the approach I'm going to go through here. So this kind of is it in a nutshell here. Our philosophy really is that a healthy soil is balanced between the blue disc, the yellow disc, and the green disc you see on the right. We see that as a three-legged stool of these different soil properties that we want to understand what's going on in each of these realms such that our stool is balanced and not weak at one place or another. And so what we talked about last week was measuring different indicators in these different soil arenas to find out if we have any limitations. Then we take that same understanding of perhaps impaired processes, if they do exist, and when we manage the soil, we can address those ideally several at a time. And I've produced a caveat down here at the bottom of this slide, which kind of gets at really the underpinnings of all this. A person would say, why would you want to increase your soil health? Well, certainly it's at the very end of things has to be money, increased profitability. We want to be more successful with our management. And so again, part one would be to identify these soil limitations. And then when we can understand it and see just what's going on, it gives us a chance to create opportunities, what I call for synergistic management. We know there's a lot of work to do out there, and we want to make sure that all our steps, all our practices are addressing, if necessary, several issues at once that need addressing. So we saw this sort of strategy last week. The main goals, now using this Cornell Soil Health Test, which you see on the right of the slide, is we want to understand these different processes going on in the soil and just how our soil's kind of doing. Seeing where these levels are at, find out where we really need to focus our attention. But then part two is where we'll spend our time today, or a lot of our time thinking, how can we then develop management strategies that are holistic, that consider all these things simultaneously to restore our soil function. And you'll notice we have the Cornell Soil Health Test Report field management sheet at the bottom left. And you see some folks in a classroom setting where we actually go through and we practice this, where we take a soil health report like you see in the upper right, and kind of break it down into 4 steps and kind of tease out as much information as we can on this part 1, so that we can develop, uh, again, a sound and satisfying management strategy. If you note the soil health report you see in the upper right, what we have here is a grower who had rye vetch 3 years ago on a field, and then 2 years of strawberries. It's a sandy loam soil, and you can see that there's a couple of where you see these red scores. We've outlined some soil health constraints, some soil processes that aren't aren't quite up to snuff that we could address with our management. And we'll come back to this at the end of the talk, and actually tease this apart and use this 4-step strategy to kind of dissect this. And I'll just give an example dissection of the information and a movement forward through the 4 steps to a proposed management strategy. Okay, so let me begin. We're going to talk a lot about organic matter today. So this is a very useful slide to kind of put things into our perspective of how we think about organic matter. So I guess this probably all makes sense to us by now, if you're interested in soil processes. But as we feed the soil this organic matter, we affect a lot of different processes, and I've outlined here in blue soil physical processes that are directly affected by organic matter additions and transformations, as well as in green, some soil biological processes that are directly affected by organic matter. And of course, these are, in fact, processes that we test in the soil health analysis. Each one of these has an analysis that gets right at these nuts and bolts of soil function. But at the same time, now we need to think also as managers. So when we say add organic matter and all these processes that are affected, well, certainly different organic matter could affect some of these processes more than other organic matter. Be the character of the organic matter, the age of it. Do we stir it in? Do we apply it to the surface? Do we grow it in as a green manure, etc.? So that's where we'll go today. Again, it asks for really holding these processes in your mind that are going on in the soil, simultaneous with the idea of managing them with very outside the soil actions by a grower. Okay, so to step back a little bit, there's some actors in this soil food web, starting with organic matter on the far left. We've got living roots and shoots, which they are organic matter. They're not dead organic matter, but they're living organic matter. And below that, we see dead substrate. So we've got the source of all our soil biological processes really is the plant. And so we want to keep in mind that these are quite variable. We have different plants that we can use in our management, different substrates that we can apply. So when we look at this table, we see that from the blue circle, the living material, we're going to affect certain living bacteria and fungi and nematodes in the soil. And same at the bottom, the dead substrate can affect this next box, this next hierarchy of organisms. But the fact that we can affect and promote certain of these organisms, or actually restrict them based on our choice of organic matter, forces us then as managers to be aware of all the influence we can have by modifying what we see at the far left, the living and dead substrates. So to kind of put that in text here, all organic matter is not created equal. I'll use this title on another slide later in the talk to kind of bring this together once again. But when we're dealing with fresh material, this could be additions like grass clippings or leaves, as well as growing cover crops, the crop itself, green, if you will, green manure. These compounds are very young and fresh and wet. When organisms attack this and when these products break down, we get a lot of these glues, if you will, that help bind the soil together. We talked about this with soil aggregate creation. Very important. Even earthworms, as they pull this material into the soil, the goo and glue on the earthworm itself helps hold soil particles together. This is really an early function of organic matter as it goes through its cycles of breakdown. This fresh material really favors what I call bacterial populations. They really take off on this carbon energy. And we've seen, of course, nitrogen immobilization, where nitrogen is made unavailable to plants because the competition by these bacteria is so great for the nitrogen that the plants can suffer. So this is just one of the properties of fresh material. On the other hand, composts, and here I mean kind of an extreme sense, compost, everything from piled up leaves, biosolids, eyeless biochar, municipal stockpiles, these sort of long-term produced composts that could be trucked and brought in. What we see here are typically more stable composts, more stable material than the fresh material. And what's happened is this composting has taken place in a pile. When we add the fresh material, in contrast, the composting, so to speak, is taking place out in the field, in situ, right in all these little micro places in the field. When we produce a compost, of course, a lot of these early processes, nitrogen immobilization, for example, is taking place in the pile, which of course we can manage to produce a net final product compost. But the idea is we're producing something where these early processes have already taken place. So the products that we put on the added compost then is more stable, which is what we get ultimately from the breakdown of the fresh material that ultimately becomes more stable. But here we're kind of short-circuiting that by doing it in a pile. But also, the last sentence here under composts, we can also concentrate nitrogen and phosphate in the compost and come up with actually a quite rich material. So we have to be aware of that as well. Okay, so we see that these biological processes then that we see in the soil are going to be affected by the type of organic matter. So how much fuel this living fresh material is in the source organic matter, and how much is already in a stable or passive condition, which isn't necessarily food for organisms, and it's more of an inert loosening compound. And of course, in our soil, we have existing soil organisms that will be affected by this material that we add, that we give to them in our management. And of course, we can affect that. And the way we affect that is with management is what we'll be talking about. But these two actors here interact through moisture, temperature, how we mix them together, or for example, tillage. The fertility level, particularly nitrogen, can affect how fast these things turn over, etc. So let's get right to that. So here it is in a nutshell. Different plants and plant debris composition is going to affect our soil biology. And this is where we need to do a lot of thinking and learning about new varieties, for example, in the cover crop arena, what I wanted to say is that I'm sure you're all aware now of these crucifers, these mustards that can have compounds associated with them that we can use to our advantage to control organisms in the soil. We've seen that any number of times now in potatoes, even in strawberries, that we can kind of manage and choose a cover crop then that has a byproduct of its breakdown that can affect the soil biology. So that's a kind of an example of how we can modify the biotic community to suppress a disease. But at the same time, when we build the soil with a cover crop, we're also keeping in mind that by making a healthier plant, we can sometimes simply outgrow um, a low level uh, a low level of disease organisms in the soil. And the way this is done is that the plant itself can produce defensive compounds that stress pests, or the plants can enhance beneficial organisms. So it's not just what we do; the plant itself is acting to um, protect itself. And I'd like to go through that a little here. So I think we all know that the plant root modifies the rhizosphere environment. So the way I like to think about it is not unlike a tree growing out in a field, that as it pulls nutrients out of the ground, it grows leaves and then drops those leaves ultimately onto that same soil. So it's in a the plant in fact does very overtly modify its environment, its soil environment, by pulling nutrients out, turning them into leaves, dropping the leaves. As those leaves break down, compounds are released that help the plant ostensibly continue to survive. In the same way, now the root does the same thing underground. So the root is producing these leaves to, if you will, feather its own bed um, by sloughing off cells around the root tip. You can see that in the photo at the bottom left. In fact, some estimates suggest that even up to 50% of plant photosynthate is used in underground to not only grow roots and the dry matter of the roots, but that the root is actually giving these cells, if you will, this energy to the environment in the rhizosphere, just like it's giving leaves in the fall to the soil above ground. So the root tip loses some of these cells, these epithelial cells, as it moves through the soil. And then as these cells die, they break open and release their contents into the rhizosphere. These cells can become food directly, or they can simply dissolve nutrients in the rhizosphere zone um, and make them available to the succeeding cells, living cells in the root tip. These cells can release compounds that are antagonistic to disease organisms in the vicinity. This is a very direct effect, but these cells and the plant is really working again in its own best interest. And the reason for this is that this root tip is kind of the pilot going through the soil. Um, it doesn't take up much water or nutrients. Where that happens is further up the root. So where I show the root section and the root hair on the top part of the slide, I'm actually suggesting that we kind of put this all together into one picture, and imagine that behind the root tip, then, as the root tip's passing into the soil, the section behind that is where these root hairs develop. These root hairs are single cells that are very effective at uptaking water and nutrients. And remember, the environment they're growing into has been affected already by these sloughing off cells of the root tip. Of course, these root hairs are what get infected by rhizobia bacteria and nitrogen fixation in a lot of the legumes. Also, these root hairs entwine with fungal mycelium, which is very important for increased capture and, again, increased surface area for capture of nutrients and water. Ultimately, these root hairs will burst and release compounds into the soil, again dissolving the soil to release nutrients, releasing compounds that again affect antagonists in the soil, and provide an environment favorable to the next root hair that moves into that zone. Okay, so I wanted to talk a little bit about just very broad sense here. A lot of interest in these different fields these days of a bacterially dominated system and fungal dominated system. And this is very broad sense, but I think it's a useful strategy to kind of keep some of these ideas in mind. So we said before that the bacteria are very fast acting. Their population changes very quickly. Um, they're at the root of the way we manage most annual field crops. We're counting on these bacteria. What they're doing then is using the last year's and previous year's organic material and mineralizing that into often plant available materials. And tillage promotes that because it stirs the soil and provides fresh opportunities for bacteria to meet fresh organic matter. So the bacteria don't move very far, but when we till, we of course add air and oxygen, but we also provide new opportunities for the bacteria. And the result of this is we get, as these processes continue, we get somewhat stable humic carbon compounds that are somewhat recalcitrant and can build up into our long-term total organic matter accumulation in the soil. But we see more of that in a more fungally dominated system, simply because when we're stirring, we always provide yet another chance to take this long-term material and have something get at it. So in a fungally dominated system, now we've kind of moved past these early stages of breakdown. And the fungus, I see them as somewhat slower. I don't mean that directly, but they're more, if you will, more deliberate in these systems in their approach to using the nutrients that are there. They take a little bit longer to associate. They're not just these organisms don't necessarily just float around in the soil. While they're in the soil, they're looking for a relationship to a plant or to the material, where the bacteria are just really more opportunists. We talked about how as these funguses develop these relationships, that they produce a wide network of mycelia that can increase not only uptake of water nutrients for the plant, but also protect the plant against outside attack by disease organisms. And since these associations are more complex, they develop a little more slowly, and they require not so much of this favoritism towards the bacterial and the rapid turnover, but in undisturbed or permaculture environments, or what we might see in a more of a berry situation. There's time for these infections and long-term associations to develop. And because of that, then we have a chance for more complex carbon sequestration into more complex compounds, what we would call humification then. And we can produce compounds that really sequester carbon over the long term, and help our soil with these with the processes that are more associated with this, what we called last week, the very dead, stable organic compounds. Right, so kind of a fun little slide I made here. I show some people collecting leaves into a leaf pile this last fall, and across the top, they simply pile them up and don't stir them. And at the end of a year, the next fall, we see that the pile is about 2/3 as high as it was the year before. Not many of the leaves have broken down. The weight of snow and rain has kind of compressed some of the air out of it, but we haven't had a whole lot of breakdown. And so my CO2 evolution out the top of these boxes is smaller. At the bottom, we show a leaf pile, the same leaf pile being collected, but we're doing a lot of stirring. So we're going to turn our compost pile, provide opportunities to move bacteria into zones of fresh material to attack and break down, and etc. And we see, because of that, then we're going to have a lot more carbon dioxide release. So as we're going to increase our respiration through this opportunity of the bacteria, and as we release carbon dioxide, we move through the soil food web to, if you will, more complex carbon compounds, the simple ones being used up by the bacteria. Um, and of course, I think we all know the model of the carbon cycle, but I put this up because I really see it as being a balancing act between the top arrow, the top orange arrow, and the bottom orange arrow. So on the one hand, we're taking carbon dioxide from the atmosphere and composing um, carbon into sugars, cellulose and starch. And on the other hand, we're decomposing through the respiration of soil microbes, decomposing this organic matter back to carbon dioxide. And it's this spinning, the rapid spinning of this, as I was saying, is what's favored by um, the bacteria. And when these cellulose and starch compounds can be more complex or just different, we're going to affect the soil microbes then that would be able or want to or rapidly decompose them. So I kind of like this sort of a concept to think about um, composing and decomposing, and what we're the kind of strategy that we're going to have to approach. And of course, when we add manures or composts on the upper right, we kind of short circuit the plant that had been done elsewhere, but then we can directly affect this carbon, cellulose, starch, whatever you want to call this complex material that then microbes are going to act on. Okay, here we go again with the soil food web. On the right, I'm showing some fungi colonizing this plant rhizosphere in a more undisturbed environment. I just wanted to use this slide to kind of introduce this idea again of we've got living and dying soil organisms. So there's really a web of life going on at quite rapidly in our soil that we're going to want to affect with our management. But first, I'm going to take just a few minutes to kind of say how we're going to look at the processes themselves in the soil so that we can apply management to those processes. So as these organisms cycle, they're basically breaking down organic debris and modifying it. And then one by one, I want to step through these other ideas. In this Cornell soil health test, we're going to look at how these organisms create and release glues and try to measure that and quantify it. On the right, I show a clump of sod dug up, and you can see the nice soil structure, the nice crumb structure. These soil particles are glued together, not only by the roots themselves of the plants and what we're talking about, these exudates from the roots, but also the organisms, the rapid turnover of organisms in that zone, helping to glue these particles together. Another thing to consider when we grow if we call this a cover crop or grow a crop, we're actually inserting organic matter into the ground when this plant dies, right where you see the roots now, as opposed to tillage, where we would or tillage of, let's say, a compost applied to the surface, where we accomplish something very different. We would mix the soil from the surface in very heavy-handedly, as opposed to growing it in place. So that's going to obviously affect our net result, and this is something we have to keep in mind as we go forward with decisions on how we want to affect our soil. And at the bottom right here, I show just a kind of a contrast in what soil looks like when we test it under the soil health test. On the left, that soil tested right in the medium to medium low range instability, and the same soil under different management of no tillage. We see that the soil is holding, these crumbs are holding up better to the action of the raindrops. Right, so these soil organisms in the soil can mineralize nutrients by breaking down. Mineralization refers to taking organic material and removing the literally the nutrients and minerals in it back into plant available forms. Plants tend to take up inorganic substances and combine them into organic forms. So here we want to go the other way and mineralize. So that's kind of the opposite of what a plant does. We want to break that plant down into ideally nutrients for our plants. And we're going to look at that. We look at this with the potentially mineralizable nitrogen test in the soil health test. We look at the rate of breakdown of organic materials to release ammonium. We mentioned before that these soil organisms can protect or infect crop plants based on what organisms are there and how we manage for or against them. We can use that to our advantage to protect our target plant. And again, this is we saw this last week. This is the soil bioassay test that we use in the soil health test. This is actually a greenhouse test where we plant. You see replicate samples here on the upper left of a soil sample where we put one of these green beans into each tube and grown it out in the greenhouse. This is this variety of green bean is called high style, and it's well known to researchers, particularly in plant pathology, because it's sensitive to a wide variety of diseases. A lot of growers or growers typically don't buy this variety anymore because it is so sensitive, but we can use that to our advantage to kind of see what's in the soil and how our management has affected these some of these less desirable players like Pythium and Fusarium in particular. And then another test in the Cornell soil health test we talked about last week was this active carbon. And here's where we're looking at the food value, if you will, or the energy, literally the energy value of the carbon in our soil. This is the what I would call the kindling of the soil organic matter. This is the easily accessible, easily oxidizable, easily available, the sugars, the cupcakes of the soil organic matter. This is bacteria would act very quickly on this sort of material, but we can measure this and say a high level of this would then suggest a high level of activity in our soil. So we can use this information as we approach our management. And we talked last week, I'll mention it again, we talked last week about how this purple potassium permanganate also can break down, not unlike the organisms, can break down and burn this soil organic matter. And so the more purple that's used up in the course of this test, it's a 10-minute shaking, the more purple color that's used up, the more available organic matter was in the sample. So it's kind of again, we're testing that flare up. We're looking for that kindling potential of the soil organic matter. Right, and here's the slide that we talked about a little bit last week about these long-term humic materials and how important they are. And literally, these are those persistent compounds that we really talk about when we say about building soil organic matter. The other material, it's just a matter of time. This fresh material won't be fresh for long. It's always on its way to being converted into something else as organisms transform it. But these compounds are kind of the end of the line. And these are the ones that, as I say, when we're building or sequestering carbon over time, these are the ones we're concerned with or that we're talking about. And that I had said we see more likely to develop in these permaculture environments, not just because of the action of this a little more fungal domination, but also because of the lack of stirring. And why these are so important directly is that these humic substances really loosen the soil. They're very light and spongy. They hold a lot of water. They soften clay soils, and they also hold chemical nutrients based on their charge, their negative charge. Oops. Sorry, I went the wrong way on that one. Slide malfunction, sorry about that. Here we go. So back to our the slide we saw before where we add organic matter. Again, these red boxes are all the different tests in the or analyses in the Cornell Soil Health Test, looking at these different soil processes. But again, I have to stress when we think of we want to understand all this, but now we want to talk about okay, different organic matter sources and qualities are going to affect these differentially. So we have to use the same sort of thinking and logic, if you will, on how these processes are affected um, through their interaction of the different realms um, of their actual qualities. Now, how could we use that to our advantage and to what we want out of um, the the soil that we're working with? Going to mention this is the Cornell Soil Health Test suite of measured indicators on the left, and on the right are the soil processes that we're measuring. Here it is, the soil health report. Again, the indicators, the value, where the blue arrow is. So these are the lab or field return values. Remember, we rate these values with our database. The lower 30% of scores in the database are scored red, and that's useful for developing what we call constraints on the far right. So if a value comes back in the lower 30% for its soil texture, then we would say yes, these functions are certainly below an optimum or room for improvement. And so this is where we kind of start to put together these different processes in the soil that we can then approach with our management to kind of get at several of these at once. As I said before, create these synergistic opportunities for knocking out multiple issues at once. And I have to mention the website here. A lot of this material that we talk about is on our website. In the training manual, very useful document, available as a free download from the website. Also, there's all sorts of information on sampling, delivery of samples to our lab, just strategies in general. And in the manual, not only does it talk about part one, the soil test itself, but what we're going to move into now, the soil management guidelines. So kind of a starting place for using the information to go forward. Right. So last week, really, we talked about the soil processes and the soil health test indicators at the bottom. We spent a lot of time on that. I introduced this idea that we can use this information in the test report then towards our soil management. But again, to use this holistic strategy, we have to understand the processes, find out which processes are impaired, then tweak our management to address them. Coming back to our soil health report from before. So this is this was a strawberry field that had, excuse me, rye vetch on it for 2 years, 3 years ago, and then strawberries were planted and grown for 2 years. It's a sandy loam soil. So the first thing we're going to want to do to use this holistic strategy is to test the soil and get the information on what functions in the soil are good, bad, and indifferent. And from this, then we can learn a lot. For example, we look at the aggregate stability, the first physical indicator. We see that it comes back at a 95% rating. It's in the green. So we've got a really nice soil structure according to the test. Yet, as we move down the list, we see that there's surface hardness and subsurface hardness. So we've got some compaction in the soil, which we'll want to take into account as we move forward. And down in the biological realm, we see we've got a decent amount of total organic matter, 2.8% for a sandy loam. That's not bad. But our active carbon, our fresh organic material, and our the rate of mineralization of nitrogen, so this breakdown of organic material into available nutrients and fresh material, that rate is also slow. Looks like our soil chemistry is pretty good. Our pH kind of at the bottom end of where we'd want it. But P and K look good. Micronutrients look good too. We've got an overall score, soil health score of 59, called at 60 in the medium range. Kind of useful for just kind of thinking in the long term. Several years later, we'll try some different management on this field, and several years later we'll sample again and see how we've affected things. So we've talked about the first step. Step 2, what might we do? Our management options. This is kind of a brainstorming strategy here, where we kind of say, well, it's certainly we'd want to identify this hard layer, find out just what's causing it, where it is exactly, what are we going to do about it? What might we do about it? I list here that we might plow it up, we might maybe use a deep ripper, we might go with a cover crop. Then to address the soil biology, while we're thinking about plowing and ripping and perhaps growing a cover crop, we want to maybe feed the soil some heavy debris. If we could get some, if that's possible, that debris could feed the soil, particularly if it's got rich biological material that would start to spin our soil food web and perhaps address our active carbon. If we use a nitrogen-rich material, again, this is as a compost, as we feed the soil directly. If we had something that had nitrogen, fair supply of nitrogen in the organic form, that would help build our population of mineralizable nitrogen critters and increase that rate. We may, in our cover crop, we would want probably a rich root residual, like we saw in the sod photo a few slides ago, where we can, maybe without even plowing, we could move this rich organic material, maybe even rich with nitrogen, right into the soil profile without plowing or bringing anything in. And that would be, for example, growing a clover or a vetch or an alfalfa. You may want to mix the disease rating here, looks pretty good. However, I mentioned these options with these new crucifers, a lot of different options out there for crops to use as cover crops and nurse crops to build the soil. But again, we want to have that in mind. Just because this test tells us we're in good shape with root health, doesn't mean we might not want to get something mixed into our rotation here that provides some suppressive capacity towards something we know that's out there or have heard about in the vicinity. So then we're going to move to step 3 and step 4. But before we do, we want to think about what we're trying to do with soil management. It's an ever more complex world where there's so much information out there that we have to continue learning, modifying what we know, asking questions, seeing what people are doing all over the world, really, certainly in our region, to learn of these latest technologies, strategies, so that we can be better, not just better growers, but as consultants and educators, better suppliers of information, and sharing this information so that we, as a group, can just all be more successful. And of course, as we try to capture new markets or work with new varieties, new cover crops, that requires us to really, I use the word transition, to be capable of applying this new knowledge then incrementally into our own minds first, then into our management, where we kind of realize that things we haven't been doing might behoove us to learn and to learn how to do. Never easy with agriculture. As you know, there's a lot of risk associated with it, and so we tend to want to be risk averse, and these changes are risk. Because there's a learning curve. So that's where the consulting ag professionals and the grower will see more and more of these liaisons being necessary, yet at the same time that we're going to see probably less and less money for state and county support for this. So we really need to be thinking, how can I get this information out as educators, but also as growers, how can I get this information in from all these different sources? Okay, and again, all this information is we really have to deal in a case by case, field by field, grower by grower basis. So even when we learn principles and strategies somewhere at a conference, at a particular grower's field, we need to take from that then the principles and say, can I use that principles back in my locale? And it's actually very exciting if you're an educator, because you kind of get to go out into the world and learn and then apply that learning, um, really without the risk that the grower has. But the grower is doing the same thing, going out, learning, coming up with new questions, of things to try, and more questions for the educator. So all this information going back and forth, it really requires considerable attention and thought, and really, if you will, creativity, to be useful, because things are changing, and that change just requires this kind of uh, a, a deliberate process of adapting to it. Right. So in our soil health manual, we've got table 5 here on page 57. If anyone has the manual, they kind of lists short-term and long-term management strategies to deal with, um, for example, limitations identified in the soil health report. But I have to mention, these are just starting points, but they do kind of get at this idea that we have a generalized model that we start from. So I don't want to say there's no answers here, but there's no recommendations here. It's information that we again apply into our model and go forward with more specifics that are germane to us, but use this just as a more of a reference starting place. So I'd like to use the first uh, circled physical concern there, this low aggregate stability. I'd like to talk in the short term how a manager might address that if that was the case. We note here that we talk about fresh organic materials, shallow rooted cover crops. We saw the picture of that grass sod um, some slides ago, showing how we could build soil structure with that. Um, of course, a rotation crop as well can provide that. When we add fresh manure or green clippings to the surface of the soil, of course, these materials will be rapidly attacked. There's a lot of fresh carbon there, a lot of sugar, so to speak, for the soil bacteria. So rapid order, they're going to turn over, spin that soil food web, and help glue our soil back together. In the longer term, again, we're going to want to be thinking about, well, how did I get into this position in the first place? It may be that we want to look at a lot of interest these days in reducing tillage, not necessarily going to what we call no-till, but there's a new term out there called low-till, where we just simply try to reduce the tillage intensity and frequency, look for opportunities to really think, can we focus the tillage right in a zone rather than across the whole soil surface, for example. We might want to think about a surface mulch. Certainly in a berry situation, we're often dealing with surface mulches that can protect the soil and provide a constant source of material that we can kind of maybe disc in or stir in a little bit once in a while to keep our soil bacteria cycling and protecting that soil structure. Okay, so again, on a broad sense, when we talk about the soil health report strategy, I've gone over this several times, and I do apologize, but I think it's important to keep mentioning this. We're going to identify um, areas in the soil that need attention, because we're determining that the processes are constrained. So now we want to combine this information with the grower's needs, skills, and available resources to come up with a strategy to deal with them. And I mentioned here the four, really our four broad sense options in management. Anything we might do to the soil really pretty much falls into one or a combination of these groups. So we can change the crop that we grow and what that leaves behind, how that organic matter affects the soil. Also the production strategy, how we manage the soil to grow that crop, affects the soil. Uh, by, again, changing varieties or to longer seasons, shorter seasons, for example, we can open windows for cover crops to be inserted into these windows. So hybrid choice becoming very important in crop rotation. A lot of documentation of what these different hybrids can do to provide even, as we mentioned before, certain chemicals in the plant that that we might want as a, if you will, as a byproduct to affect our soil environment. And of course, cover crops themselves, what an easy thing to say. We need to grow a cover crop, but that that only puts the book on the table. It still needs to be opened and read. And this is where it's a brave new world. A lot of information out there. We just need to really be on top of what all these options are. And we'll go through that in a moment. Organic and chemical amendments. When we manage the soil, certainly I mentioned a couple times, we can bring material from other places in and apply it to our soil surface, either as organic or chemical, and work that in or leave it there. And of course, we typically work it in with tillage and soil stirring. And so we always have options to manage the soil that way. So we'll go through those one by one. The crop rotation, suppressing disease, smothering weeds, all these, again, kind of broad sense caveats of really an agronomic approach. When we rotate crops, we can, again, put different material there or not there at different times to affect our soil biology. Different cover crops, same idea, but there's different windows for different cover crops. And again, they're going to have different qualities, the different cover crops. Not only when they're because of when they're grown, but what they bring to our the organic table, the palette. Organic and chemical amendments, my gosh, manure and compost. So easy to list them, but the diversity of manure and compost and green manures, how we might use these. Again, it's very easy to list these, but there's a lot of information behind each of these that we'll discuss in a moment. But we have to be aware that it's going to take creative thought and movement into the page beyond this page to make our best decisions. And of course, I mentioned crop residues themselves, biochar, then more conventional fertilizers, pesticides, herbicides that we can also add to the soil to manage it. And of course, tillage. I mentioned the low till and no till. Tillage can break up or prevent compaction. And there's lots of new equipment out there that allows us, I call it innovations in disturbance, different ways we can stir the soil, different shapes of shank, everything from different shapes of shanks, coulters, etc. That can be very useful for us. Okay, I show here on the right crop rotation on organic farms. That's a fairly recent publication available through the SARE website, sare.org, for a very low price, also available free as an online download. This looks like it's for organic farms, and it is, but that's not to say that there's not principles in here that are useful for any grower. As you know, organic growers must be very cognizant of crop rotations to protect their or to affect their soil biological populations, because they don't have the literally the luxury of coming in with a very specific chemical that has very specific chemicals that have been developed for us by chemical companies to kill or manage very specific pests. So the organic perspective here is very useful towards as we strategize towards understanding of how we can affect our soil biology. Right. So the different crops suppress or break disease cycles. There's by growing crops that take more or less time, we open up windows where you can grow a cover crop, for example, to smother weeds. Ideal crop rotations best utilize our land, equipment, and labor resources. Allow us to grow crops that people are clamoring for and paying money for. And of course, rotation with sod, I had to mention this. In a when we talk about crop rotation, it's important to consider taking a field not out of production, but putting or part of a field into sod for all the wonderful things it can do um, to rebuild the soil very dramatically and drastically. Wanted to talk a little bit about cover crops. Again, wonderful book out there, Managing Cover Crops Profitably, available for free to download at the website below or very low cost. And this gets at the culture and aspect and um, qualities of a whole suite of cover crops for the whole, certainly for the whole Northeast. And this book actually goes further south and a bit into the western state, in the Midwestern states, talking about all the different um, effects and culture of cover crops. But again, different cover crops will have different qualities, more or less of them, of some of these listed features here. So if we had an issue of that we wanted to, for example, add nitrogen with a cover crop, obviously we want to choose in the legume family, but the idea here is we want to really be wise and clever and choose well. But there's a lot to learn. We can't. This isn't just an off-the-cuff decision. I'm going to plant a cover crop and now I'm done until I plant it. No, that as I say, that book needs to be opened and takes a lot of discussion, a lot of research, a lot of questions to be asked and answered by the grower and any sort of consultant, so that we can maybe even with our cover crop not only address some of the things we see here, but do more than that simultaneously. And again, some cover crops do several of these singularly circled factors at once. All right, there's even a online cover crop decision tool. I've got the website down at the bottom. This was developed by Thomas Bjorkman, a Cornell researcher up at Geneva, where you can actually type in a management goal. So what he says, the reason you need a cover crop. So from that last slide, if we wanted to enhance one of those particular features, if we're really looking to affect one particular aspect of our soil very powerfully, we could type that in here, and then the decision tool would also ask us, well, when is your window? How long is it open? And then it would give you different options of cover crops that fit that need. But again, I'm going to go over all four of these bullets here. Note that each option tested in such a strategy is always an iteration towards a final decision. I had to read that. It's this idea that we need to kind of go through it in our mind and say, does this work, or how well does it work? Because we can always modify that a little bit towards what we need, and that we have to keep that in mind. Lots of success through sharing success stories. When we see a grower with a particular situation in an area that kind of comes up with a cover crop combination that has luck with that. We hear about it at a field day, for example, or we see something. We can think, now, how can I modify that or use that for myself as a starting point? We've seen a lot of great, we've heard a lot of great success stories built out of success stories. Growers often start small, and we recommend this, using strip trials or half of a field managed one way or another way to kind of learn on their farm what works or what they find just won't work for them. Also, as we enter into new worlds of cover crops and different rotations, we can also introduce new pests and problems that we didn't have before because we weren't using these strategies. So again, we have to be vigilant and ready to learn whatever's necessary to move forward. Right, now I want to talk a little bit more about this managing of organic matter. Again, I put a book by Harold Van Ness and Fred Magdoff in the upper left corner, this Building Soil for Better Crops. Um, Available free online, very low cost if you want to own the book itself. And what this book talks about in great length is how to balance this idea of we want to decompose organic matter to release nutrients. We want the organic matter to break down to, as I've mentioned before, glue our aggregates together. We want to feed the important soil biology. But at the same time, we also want organic matter to accumulate for all the important reasons that we want organic matter in our soil to store water, to retain nutrients, to loosen the soil. Carbon storage. So really the only way to be successful at this is to literally keep adding organic matter, because as we decompose it, we have less, and we can only accumulate it by growing or adding more. So we need to kind of tip this scale the other way, so we're kind of accumulating on a net basis, or I should say a gross basis, even though at times we need to decompose some of that organic matter. So again, we're going to have to feed the soil with different materials and regularly. And the more diverse organic matter we can add, the broader a base of organic activity we have in the field. And again, I mentioned compost, cover crops, these crop residues, adding leaves, biochar. When we add these different materials or grow these different materials, remember that we're going to accumulate a different kind then of soil food web response to this different food source. And that's really ultimately what we're managing for. Here's this slide, all organic matter is not created equal. I've talked about this any number of times now, but if we look at this pyramid, when we add a green matter or a green manure as a cover crop, we're adding these sugars, these lighter, less complex compounds that the bacteria are very hungry for. When we add composts, or when we're dealing with composts, we've moved further down the pyramid already in the compost pile into these more complex, longer lasting materials. And again, the different types of organic matter are going to affect the bullets you see at the right. Some of them will affect soil aggregation, particularly the green matter, but different green matter will have different effect on the soil itself. We've all heard of, for example, buckwheat having a particularly profound effect on soil aggregation in a very short time. So we need to keep that in mind. They're easy to list as bullet points, but we need to think about what exactly will do that to affect our management. Right, and also this is also something we see in Harold Van Ness's book. More is not always better, so we have to be careful, particularly when we apply compost and manure on the surface of the soil. It's very easy to over apply and cause leaching and denitrification concerns. In vegetables in particular, we don't want too much vigor from too high supply of nitrogen or other compounds or other nutrients in the soil. Also, over application of compost might keep the soil too wet. All important considerations to always work within. If I can move down to the last bullet point, to kind of start from what I'm calling an industry standard. We don't want to go overboard with these application strategies. But we want to kind of start from a safe place and then modify from there as our crops and our field and our material requires. And keeping in mind also that, particularly, I give this wine example here, how some crops, grapes grown for wine, actually we don't want this great vigor, and we want to be very careful of what we're adding. Just kind of a, if you will, an over extreme example of really thinking ahead towards what we're doing and make sure that that is indeed what we want. Oh, right, back to our stirring of the soil. Just again, to speed up these bacteriological processes. Um, the our organic matter is going to mineralize faster when we stir it. And the way we do that stirring so often is with tillage. Um, this is a slide showing tillage addiction. So I'm going to go from the upper left clockwise around where we till a soil. We see it being completely inverted. This is a sod. It's being plowed a little wet, kind of show the kind of a worst case scenario here of soil tillage. Nonetheless, we're always involved at kind of some aspect of this. So after we flip the soil over with this tillage, moldboard tillage, now we've got cloddy soil that needs to be made into a seed bed or a planting bed. Then typically we come across the field and pack it down, firm it up, but then we're set up for potential crusting. The soil is unprotected, it's bare. This is a kind of a worst case scenario, but I show this to make people realize that, or not forget, I should say, that tillage is a very powerful tool that we shouldn't take lightly. That when we apply tillage, we're really affecting a lot of soil processes, not just the physical things you see here, but the organisms are going to be literally tipped upside down, and they're going to respond to that in a way that may be useful to us or maybe not. So we want to keep that in mind. We see a lot of focused tillage through this sort of ripper strategy. Instead of the last slide, that was a full width moldboard plow just flipping over the entire soil surface. These rippers, I think everyone's seen these, have a rather narrow shank, typically an inch wide. This is shown set up for row crop production. However, the idea is that we focus the tillage really right in the vicinity of this rip shank. We don't flip the entire surface over, and we end up really just kind of plowing in a narrow zone. Here's kind of what that looks like then in the upper left. We've got kind of an undisturbed surface for most of the soil surface, but then our rip shank can go at a variety of different depths, whatever's necessary to break restricted layers up. And we're left with, if you will, a no-till environment between the rips and a tilled environment where the rips are. Very handy in approaching certainly a bedded situation where we're almost forced to do this. But keep in mind that by focusing the disturbance, we can kind of get the best of both worlds and allow us. And this is of course a field crop example, but we've got then a hybrid system where we've plowed where we're going to plant the seed and have a small plant. But then as the plant grows and explores new areas, it'll actually be growing between the rows into a virtually unplowed or no-till environment. So we've got a plow zone and an unplowed zone, and so we can take advantage of the season. If it's wet or dry or periods of wet and dry, the plant has areas where the root is where the soil will be drier or moister because of this hybrid scenario. So I really like this sort of slide to show how when we change our management a little bit, if we look at the bottom center picture, we see on the right the conventional, if it's hot and dry, our entire soil surface is going to dry out, for example. Where with a restricted width tillage, where we focus our tillage, if we get a lot of hot, dry weather, we're going to have a little bit more water storage underneath all the debris between those ripped areas. Right, and I list here some of these tips for transitioning. That's one of my favorite words to reduce tillage crop production. The idea is that we're trying to transition to less tillage. So we're thinking about, do I need to do what I've always done? Is there a new method out there? Or I've heard of this new method. How do I transition my field into using it? And here's some of the ideas. So first of all, soil compaction is a question of soil being dense. To alleviate that, we want to loosen the soil. Again, this is with the iron in the field as opposed to using plants to do it. But we can focus our tillage to specific zones. in particular, for example, a wheel track zone, maybe the only area compacted in our field is where a particular piece of equipment went. Well, it might behoove us to focus directly on that rather than attempting to loosen or till the entire field. And that's where we can also, when we put this in concert with the green text, I guess number 4, we can feed the soil food web with our organic material and combine that with our tillage and say, wait, this is how we can accomplish more than one of or a second and third objective simultaneously in my management strategy. Of course, then there's longer and shorter term strategies for that with the cover crops, rotations, adding organics, reducing tillage. We listed those in the toolbox. When we can build the soil and reduce our tillage and build it with rebuild our soil structure, the soil can become more resilient to weather extremes. And I have to mention at the bottom that when the soil is in this, if you will, healthier condition and in better balance, everything seems to work better. This idea of applying reduced tillage to huge effect, positive effect, is enhanced when the soil is in good condition. Here I just wanted to show how there's a synergistic effect to our management. So we see soil health on the y-axis and years across the x. If we apply if we modify our management with finding windows for different cover crops or reducing our tillage, individually, if we apply these managements, we can increase our soil health. But if we can creatively put them together and combine them with an overall holistic plan, we tend to see faster and greater overall response, what we call an additive effect of putting these positive management approaches together. Kind of a synopsis slide here showing the a soil health report on the left, the one we're going to talk about in just a minute as we go through the final management stages of interpreting this. But we do want to remember that the soil health tool, the soil health report, is a tool in a whole program of developing solutions to farm, field, market issues. What we're talking about here is that we have this framework for guiding our soil management using using a process-based approach, but in the context of the grower's needs and desires and limitations. And I've mentioned this several times, but this idea of there's always forever on now, there will be more and more information available to people in the field in terms of strategies and technologies other people are using, and that's going to require that we stay abreast of this so that we can do our best job to not fall behind the rapid pace of soil management these days. Also, I had to put this in as a this idea to illustrate this idea of creativity. We really like this kind of going from a closed. I like going from this idea of a closed. A closed mind that says that this won't work here. So we're dealing with some extension folks up in Vermont that said, well, we can't put cover crops after our corn silage up here. Our growing season is too short. And some specialists up there said, well, wait a minute, let's work with some of the growers and see if we went to for example, a shorter season variety, opening up a window of time at the end of the season for a cover crop. They tried it. They went with a 10-day shorter corn variety for field corn, and allowed them to actually be successful with establishing a rye cover crop. With these just a few extra days, gave them quite a heads up on using the tried and true older season varieties. So it just goes. I just wanted to show with this that sometimes there's with a little bit of risk and creativity, kind of put together and try something as a little trial, we can come up with something that's useful to perhaps a lot of people. Okay, so I'm running over here, so I'm going to go quickly through these last two steps, step 3 and 4 of our soil health report here. We said this was a 2-year strawberry field coming out of rye vetch 2 years ago. This grower is willing to experiment with different cover crops. He's considering transitioning to organic because the market value. He does have a dairy farm nearby. Just a little bit of background on who exactly this person, or who specifically this person is. So step 3, we say we want to apply this the soil process information that we have in front of us with the situational opportunities. Where is this guy? What's he up to? What does he have at his disposal? So we listed here step 3. This grower does have access to a grain drill. This local dairy nearby has a deep ripping machine for possible decompaction. This dairyman will bring his big tractor and this ripper to the field and a person, and we'll rip this up agrees to rip this up for $100 per acre. So this is an 8-acre field, so for $800, or to do half the field, this dairy farmer is willing to come over and experiment with that. The grower again is interested in cover crops, so he attends a workshop. Go to step 4. Oops. Huh. It's there. One moment. Here in step 4. So putting all this together, sitting with the grower's come up with a management strategy for this year in the spring. He's going to plow the field up and seed a rye vetch mustard mix. And he's trying to get a sod forming cover crop there with the rye, the vetch to supply nitrogen, as well as a deep taproot, the mustard for some disease suppressive compounds. He also wants to try a strip trial of Rudbeckia or switchgrass. He learned this in his cover crop class. These crops have good cover crop qualities, and the organic debris from them is useful in maintenance of soil organics for strawberries. This grower is proposing to then flail mow and deep rip during 2012, leaving whatever in any of these organic growth systems here, including a strip trial. He's just going to mow that and leave it on the field, so everything's just going to be grown and left in place. He may even consider a different cover crop if one of these doesn't pan out, and then he'll decide based on how the soil looks as the season goes on in 2013 if he'll continue this cover cropping or perhaps go to a cash crop like sweet corn. So that's the end of the presentation. Just listed here some of those ideas we've been over a couple of times during this talk. The soil health test, identifying these constrained soil processes, kind of the beginning. We're going to keep those in mind as we think what might this grower do, or what might I do to get at these constraints. What can I try to see what will really work? Adapt many sources of information. Ultimately, building the soil to increase this resiliency to extreme weather we're going to continue to see. Okay, and with that, I'll say thank you and open the floor to questions.
And I just would encourage anybody that's been participating to use your chat box in the lower right-hand corner and type in any questions that you may have so that Bob can respond. Glenn had a comment. His comment was resiliency to extremes. Interesting given increase in intense rains. I don't know whether you have any comments to that, Bob?
Right. Well, I'd kind of mentioned this at one point. Anytime, when a soil's degraded, it's already down, and then when we get extreme weather, then it gets kicked, so to speak. But when we can build the soil and certainly protect the surface, but that surface can also be protected, if you will, internally by getting organic matter to break down in between these particles and help glue them together. Again, building our soil structure in particular really gives that's the foundation, if you will, the house. They talk about soil structure being the house for the soil microbes to live within. We can kind of build that, or as we build that, we're just making a more stable environment for whatever is thrown at us, be it heavy rains or drought. Um, from what we've seen, at least in this area, this winter, it's been very different than what we're used to. But I think we can get we should get used to non-typical weather. Um, so that said, we're going to have to modify our management so that non-typical weather isn't as hurtful or damaging as it could be. So that's going to take, again, that sort of creativity, that kind of hedging the bets with management to get at processes that are going to leave us in a um, better position to weather, pardon the pun, the weather that comes.
I do want to mention, in your discussion you were talking about compost, and I think people equate compost with a lot of very positive attributes. And I will say that, one of the things listed there was municipal compost. I would just encourage people to be a little careful with municipal composts. You should really ask for an analysis and a heavy metal analysis of those kinds of products. And even the heavy metal analysis can be a little misinforming. We had a composting facility in Syracuse area, and Syracuse has some industry in it. It's not just a residential community where you'd expect things like copper in compost made from those materials, but it had some light industry in Syracuse, and they used lime at, I think it was close to 1/3 of the dry matter weight of the material as, let's say, an adjuvant to help processes along to reduce the pathogen level down. And in the process, it does tie up heavy metals. And when they did the testing to test what the heavy metal levels were, they were not really available at really super high pHs, which this turned out to be. And a homeowner could be fooled and purchase, or a grower could be fooled and purchased material and thinking that it really didn't have any heavy metals in it,